Based on what you have learned about the observations made by Sumner, Northrop, and Moses Kunitz, do you think all enzymes are proteins? The answer is false. So this is false. The answer is no, because we have what we call ribozymes or catalytic RNA molecules. So we have these two guys, Sidney Altman and Thomas Scheck, who discovered ribozymes. So they were working on the mechanism of splicing. If you are familiar with splicing, it's the transcriptional processing of your RNA. So after your RNA gets synthesized, it undergoes some transformations. So there are regions that are cut off and regions that are connected back to each other. So this is splicing. And the molecules that were to for responsible for catalyzing these reaction were found to be RNA molecules. So initially, they discovered that the molecules responsible for cutting the RNA are a combination of protein and RNA. But when they tried to remove the protein, the remaining RNA was still able to perform splicing. And that is how they were able to conclude that these RNA molecules can catalyze reactions. And for this, the two guys received the Nobel Prize in Chemistry back in 1989. But in this discussion, in this module, we will focus on protein enzymes. We will reserve your RNA and DNA stories later. So your RNA is a single-stranded molecule, and unlike DNA, which is double-stranded, it can assume a structure like this. So you can imagine it's a straight chain that can fold on itself. So if DNA has this fixed double helical structure, the structure of your RNA is actually more diverse because of their ability to fold by itself. So probably this is the reason why some RNA can catalyze reactions, because they can also assume several three-dimensional structures. Now let's go back to protein enzymes. So is this statement true or false? Only the tertiary structure of enzymes is important to their activity. So if you recall, there was a guy during the time of Northrop and his colleagues. He wrote a treatise entitled Enzymes, and he suggested that there are the thing responsible uh, or the factor that allows the enzyme to interact with the substrate are weak interactions, right? And that is actually the basis for this statement that the structure of the enzyme is important to its activity. But in fact, it is not only the tertiary or the three-dimensional structure of the enzyme that is important. All levels of its structural hierarchy are important, because you have the primary, which is the amino acid sequence of your enzyme. The primary level or the primary structure dictates how your enzyme will fold into its three-dimensional structure. The secondary structure will tell you if in this region you will have alpha helices, in another region if you will have beta sheets, and which of these regions should comprise your the region to which your substrate will bind. Of course, we are convinced that the tertiary structure is important, but keep in mind there are enzymes that are multimeric, so their quaternary structure is also important. In fact, there are some multimeric enzymes each domain or each subunit of these multimeric enzymes perform a specific step in the reaction, and they perform the steps, the sequential steps, according to their order in the quaternary structure of the entire enzyme. Now, each enzyme has what we call an active site. The active site is the portion or the region of your enzyme where catalysis occurs. So if this is where the catalysis happens, then this is where your substrate should go. So here we see carbonic anhydrase. So this is an enzyme that catalyzes the conversion of carbon dioxide to bicarbonate. And this is actually important because one of the buffer systems in our blood is the carbon dioxide bicarbonate system. And this carbonic anhydrase then plays a very important role in maintaining the pH of our blood. Now here, we see in the structure of, or this representation of the structure of carbonic anhydrase, we see a pocket-like region, right? This is where the reaction takes place. Now, if you notice in the structure of carbonic anhydrase, there is a sphere inside the pocket, right? This is actually zinc ion. So some enzymes possess these cations, especially in their active sites. So if you have such inorganic cations, we call them cofactor. So cofactor is an inorganic ion that assists in the catalytic action of an enzyme. So again, in this example, we see zinc ion in the active site of carbonic anhydrase. And if you look here, we see that this zinc ion is fixed in place through three histidine residues. So probably it's the nitrogens from the imidazole side chain of histidine that interacts with this zinc cation. It's because the nitrogens have lone pair. Now the zinc interacts with a hydroxide ion. In fact, this is considered a bond, okay? Now this hydroxide ion that is fixed in the active site is the one that attacks the carbon of the carbon dioxide molecule. Now, why does the active site have to keep the hydroxide ion in place? That is actually one of the ways by which your enzymes catalyze a reaction. Okay, so they fix some molecule that is important in the reaction in place. So imagine if these hydroxide ions are just freely floating and then they want to react with carbon dioxide that is also freely floating. Then it's just all about chance encounter, right? But if your hydroxide ion is fixed in place, then there is a higher chance of this hydroxide ion being able to encounter a carbon dioxide molecule. So once the oxygen attacks the carbon of carbon dioxide, then a water molecule enters the active site, or probably it is already there in proximity to the reaction taking place. Again, this water molecule enters and interacts again with zinc. And this pushes your bicarbonate away from the zinc cation. So you see here a bicarbonate ion is released, and then From this water molecule, it releases another proton, and your active site is returned to its original state, ready for another round of reaction. So these cofactors are important basically in holding either negatively charged molecules or electron rich molecules. Now, we also have what we call coenzymes. These are organic or metallo-organic molecules that aid in the catalytic action of enzymes. A classic example of a coenzyme is NAD, or nicotinamide adenine dinucleotide. I'm sure you are familiar with this molecule. This is important in the synthesis of ATP, right? Another example for this one, coenzyme A. I'm not sure if you've heard of this, but if you notice in this table showing some common coenzymes, each coenzyme has a specific function. And most of these coenzymes basically support or assist in the transfer of a certain chemical group. Transfer means you have a certain source of this, for example, hydride ion. So you have a source of hydride ion. Then your NAD assists in transferring this from the source to your substrate. Notice also that these coenzymes have dietary precursors. So, if we ensure that we have enough intake of these precursors, then we don't have to worry that we don't have enough of the coenzymes that are important in the function of enzymes. Now, if your cofactor or your coenzyme is covalently bound to a certain amino acid residue or even to more than one amino acid residue, we have a term for such kind of cofactor or coenzyme. We call them prosthetic group. So it doesn't have to be a covalent linkage, but if the cofactor or the coenzyme is tightly bound, so the interactions are very strong, then we can also call it a prosthetic group. So one, I think you are familiar with heme. It's a very common prosthetic group. So heme is not only involved in the transport of oxygen, but it is also involved in the production of your ATP. So this, the blurry structure there is your cytochrome C. Your cytochrome C is one of the complexes found in your mitochondria responsible for the electron transport chain. And in your cytochrome C, you will see two heme prosthetic groups. And in addition to this, it also has two cofactors. So you see here copper ion and copper ion. So these four create a channel through which electrons can move. It's not exactly electrons moving through the channel, but these cofactors and prosthetic groups pass on the electrons. Okay, so that is all. Those are some terms that you need to know regarding your enzymes, specifically those molecules that help enzymes perform their catalytic action.